Assalamu alaikum wa rahmatullah. Welcome everyone to our second day of your Guide to Health Research webinar. So yesterday we have discussed a brief introduction about research, in general types of research, and in specific the quantitative research methods, in addition to the critical appraisal. So today we'll start by discussing the various types of quantitative sampling and which one present the most rigorous approach to be applied in health research. So we will focus on developing a definition of the quantitative research projects, understanding the principles of setting up a clinical research, and discuss the key agencies and their roles in clinical research. In addition to discussing the regulatory steps required in clinical research with the key personnel roles in any research application. So, quantitative study was defined according to a researcher by explaining the phenomena by collecting any numerical data that are analyzed using a mathematically based method, in particular using stats such as the ones we have explained yesterday using R or SPSS. So research conducted with a human subject or on a material of a human origin, such as tissue, specimens, and cognitive phenomena, or for example, for which an investigator or a colleague directly interacts with the human subject. So excluded from this definition are the in vitro studies that utilizes human tissue, which cannot be linked to a living individual, meaning that of a deceased individual. So an important notice in research is causality. And one of the main purposes of a quantitative study is to detect the causal relationship between any variables. So a causality means detecting an effect or an impact on a condition that has caused or occurred by a causal relationship. So in a patient-oriented research in which this type involve a particular person or group of people, or even we use materials from human. These can include machines of human disease or the therapeutic interventions, clinical trials, or development of new technologies that can be applied or can be applied later on in research. So a hypothesis is a research question, which is the thing that we want to examine or assess by carrying out this specific research project. And there are, of course, phases of clinical trials, including phase 1, 2, 3, or even phase 4. In phase 1 of any clinical trial, these are healthy volunteers or patients, but it has a small sample size. In addition, it examines the initial safety, the tolerability, and the pharmacokinetic with or without assessing the effect of pharmacodynamics. While in phase 2 of quantitative studies, it also includes patients which focus on proving a concept in small cohorts, including phase 2A, or even in a larger population, which occurs in phase 2B. So in this phase, we can confirm the safety, the tolerability, and the PK in a patient and exploratory efficacy of studies and research. While in phase 3 research, it also includes patients, but it compared the new technologies and the standard treatment that was already being used. So phase 3 is conducted to assess the safety, tolerability, and the PK with the efficacy in large cohorts. And in this phase, it is usually full randomization and double blinding, but randomization and blinding is not exclusive to this phase, but rather more common in this phase. Where in phase 4, this is the post-research phase, which is the marketing and after the studies that have been ended. So some of the common clinical trial design and quantitative studies include the double-blinded, the randomized, and the placebo-controlled study, all of which can be applied in any type of clinical trial, depending, of course, on our hypothesis and our available facilities. So there are some trials that are conducted to demonstrate the superiority of the issue that is being examined or the reason why we are conducting this research. This includes a placebo-controlled double-blinded study, and it has been found unethical to withdraw any effective treatment from patients. This was developed according to the Declaration of Helsinki after the development of this trial in the late 1960s, which we said yesterday that based upon them, the good clinical practice was developed in regard to the ethical issues. So a placebo-controlled trials provide the best evidence to substantiate the claims that a drug safety and efficacy can treat a specific disease. So if we want to prove the efficacy of an intervention, which we are carrying the research for, we should do a placebo-controlled study. So as we mentioned in phase 1, this has the evidence of correct dose selection, the receptor occupancy, and the target pharmacotherapy with the PK and the PD. So in a question-based approach in research, this can include the conduction of either on a healthy volunteers or even on a patient participant. This will depend, as we mentioned, on our hypothesis. And in the healthy individuals, we can have a disease initiation event that can be developed in cohort studies. And we can also assess the markers of the disease itself or even of the treatment efficacy. So the markers feature can help us in deciding the specificity of the medication we are conducting in the clinical trial, the sensitivity, the positive and the negative predictive values, in addition to its sensitivity to the change and to the treatment adaptation. Also, it can help in assessing any side effects or risk factors associated with this intervention. So in clinical studies, the protocol focus on a question-based approach. These must have the objectives of assessing the safety, the tolerability, the PK and the PD of the medication that we are conducting the trial for, and its efficacy. So this should be a regulatory study, meaning that it should be controlled in every aspect, in specific because we are administering a medication in this type of research. And it can be exploratory in the methodology part. This will be in the fundamental basic research, as we mentioned yesterday. So, of course, there are types of randomization. These can be double-blinded, single-blinded, open-label crossover, parallel arm, or adaptive design. In addition, it can include placebo control or active comparators with the patient stratification sampling and also calculating the sample size. And in every step of any research, we should always document any data that we have, and we should ensure that we have the required document to carry out an ethically approved research. So we should document the protocol of the study, the informed consent that we have obtained from the participant, in addition to the patient information sheet, and the clinical investigator brochure with the integrated research application system, and any other materials that we have used, including questionnaires, flyers, or any other type of recruitment method we have used. So the key roles in any clinical trial include the sponsor, for example, with adding the required documentation under each person responsibility. These can include the contracts, the submission, the initiation meetings, and the study monitoring. And in every study, there should be a team of a principal investigator who is in charge of retaining the overall responsibility of the study. And also, they can delegate the action to the clinical trial coordinator any research physicians or nurses, and any statisticians or technicians. So when we conduct any trial, what are our considerations in regard to the quantitative sampling techniques? So we should focus on the research ethics committees, which can protect the human rights of the participant, and it will give balance between the independent view of the research in the assessment and in the monitoring. Also, it is required by regulatory bodies to have an ethical approval and prevent scandals in any biomedical research with the fundamental focus on the participant safety in all steps. So who are the ethics committee? These include any persons who are in charge, including the funders, the clinicians, the pharmacists, and any other interested parties. Also, some of the other in charge committees are the HR, which are assessment of the governance and legal communications. It can also replace the local checks of legal compliance by each participant organization, and it can allow the organizations to focus on capacity and capability. So in the current research educational systems, it can have simultaneous processing with a timeline of the application processes and even writing clarification or approval and amendment according to the study that we are applying for. So if the patient of our research was involving human patients, participants, rather than a theoretical study, then we should always obtain approval from the relevant bodies. And of course, as we mentioned, depending on the type of the sample that we have, this will depend on the approval that we would be required to obtain. This can be a facility approval, funds approval, depending, of course, on our research. And if we were, for example, using gene therapy, we need to check for the advisory committee involved and the healthcare products regulatory agency for any medical devices and clinical trial authorization. So before we apply for any approvals, we need to plan our research, including the identification of sites, the potential participants and resources, and consider any assistance that is required with ensuring sufficient funding that is available. So the feasibility process will include that all studies taking place within the ICHN can or need to be reviewed for their feasibility. This should include the local feasibility, and which is the process by which the local research team can consult with the relevant division of the research management team before any organizational readiness is issued. And for local feasibility, these can include the supporting services, including pathology department, imaging department, etc., as they can provide the capacity and the capability to undertake the test or the investigations, or even dispense the medication in the manner of the study protocol that it requires. So for the progresses of the safety report, there should be an annual progress report to the approved committee for the research. These should be conducted on the anniversary of the approval that was obtained and should be conducted annually until the end of the study. And a copy to the sponsor should always be emailed with any updates or progress in the study that occurs. So the development safety update report can sometimes replace the annual safety report and should be sent out in the anniversary of the first international regulatory approval until the end of the study. Also, a copy should be sent to the sponsor in this case. And a declaration of the end of the study should be emailed to the approval committee within 90 days of the end of the study. So in quantitative studies, as we mentioned earlier, the sample size and the power calculation can be critical component of the study design, and therefore should be calculated very specifically from the start or from the designing phase of the study. So according to the CONSORT guidelines on reporting of clinical trials, it says that ideally a study should be large enough to have high probability or power of detecting as statistically significant of a clinical importance a difference of a given size if such differences do exist in fact, meaning that if a true effect was present in our study, then the sample size is large enough to detect that change. And yet, there are countless review and methodology papers in recent decades that have commented that too many research studies, experiments, and clinical trials are performed with sample sizes that are even too small and too small to be reliably detecting the importance of the effect, as there are only 36% of clinical trials that have negative results and 80% of the power to detect about 50% of the treatment effect. While in another review of about 250 studies, the median statistical power was only 21%, therefore implying a shortage in the calculation of the study sample size power without being able to detect the true effect in any study. So, what is a power? The statistical power of a test is the probability that it will correctly reject the null hypothesis when the null hypothesis is in fact false. That is the probability of not committing a type 2 error that we discussed yesterday or making any false negative decisions. So the probability of committing type 2 error is referred to as a false negative rate beta, or is the power that is equivalent to 1 minus beta. Therefore, the power is the probability that we will get a statistically significant result if the assumption that we make about the magnitude of the study or the true effect are correct. So the power of the study, of course, will represent the strength of our sample size and the true detected effect. So there are many factors that can influence our chances of getting a statistically significant result. This will depend, of course, on the magnitude of the differences between the groups that are being compared. As the greater the differences is, then this will mean more chances of detecting a significant effect. Also, how variable is the difference between the groups that is being compared is very important in the sample size, as greater variability will mean less chance for significance. And also it will depend on how many subjects we have to make the comparison. These will include more subject, meaning more chance of significant results being detected. So a reminder of what we discussed yesterday, the sampling distribution of a test of STAT under the null hypothesis that is centered at the zero when the null hypothesis implies zero is how we analyze our data in any quantitative research. So when we perform a statistical significant test, we will compare what we see from a sample with a theoretical sampling distribution. And by convention, we usually declare a test result to be statistically significant if our observed value lie beyond a sided area in under the curve of a 5% null hypothesis, which is a p-value of minus or 0.05. Therefore, if our observed test of STAT value lies beyond a certain critical cutoff point in the null hypothesis, then the distribution, we would have a statistically significant result. And this would take this to be advisor or an evidence that our data were not consistent with the null hypothesis because our result was not zero but rather a higher or smaller number. So the power calculations, if the null hypothesis is not true in this case, as the true group means, for example, are actually different, then we can use something called the alternative hypothesis. In this case, the sampling distribution of our chosen test of stat equals the difference between the means that would be centered around true difference. For example, 20 units in this sampling distribution. So we may see get a result of significant of test of stat under either of these, the null hypothesis or the the alternative hypothesis. This will mean that we have simply seen an unusual sample from a population in which the null hypothesis is actually true, or because our sample was not representative of the population. And this will be true in the case of the sampling distribution or the sampling method that we have conducted, and if it wasn't developed correctly from the start. So an observed test of stat that is located at point X is more consistent with the alternative sampling distribution than it is with the null sampling distribution. And in a quite large populations of the alternative sampling distribution, this can lies beyond the critical cutoff point in the null distribution value. And also an observed test of stat located at point X is more consistent with the alternative sampling distribution than it is with the sampling distribution. So in a quite large proportion of alternative sampling, this can lie beyond a critical cutoff point in the null distribution. So our results will depend on our sampling method and if it was actually representative of the population, and also on our detected effects. And in this proportion, the alternative sampling distribution for some of tests of stat, which lie beyond the critical cutoff value, will have the power of the test comparing the null and the alternative hypothesis. So statistical power, as we defined earlier, is the probability that it will correctly reject the null hypothesis when it is actually false, and meaning that there is actual effect present in our sample. So the degree to which the two possible sampling distribution overlap, and hence the power, is influenced by the differences in the location of the two distributions and the width or the variability of the two distributions. And in turn, it is a function of the underlying standard deviation of the individual measurement and also the size of the sample from which the test of stat was calculated. So the overlap will be decreased, then the power will increase with an increased delta between the centers of the two distributions. The delta point that is drawn in this figure here we have on the side. Or even the power will be increased for a fixed delta by increasing the sample size, which will decrease the width of the standard error and of course of the sampling distribution. So this is an equation that we can use for the standard deviation error. And the sample size is what we can manipulate to change the degree of overlapping between these two graphs. So as we mentioned earlier, the magnitude of the difference between the group being compared will usually be fixed. And for all of these points will depend, of course, on our sample size calculation in regard that the degree of the variability that we want to detect, or the difference, or even the sample size that we can recruit in our study. So in summary, for sample size power calculation, in practice, we usually make assumption about the expected effect size and the relevant underlying standard deviation. With these being fixed, we can also calculate the sample size that is required to give us high power, which will indicate minimal overlap of the sampling distribution. And fortunately, for most sample or simple calculations, we can use any statistical theories which will describe the mathematical shape of the sampling distribution. This will help us to work out the mathematical relationship between these quantities or between the questions that we want to ask for our study. And many exact formulas are complex to calculate, and therefore there are various approximate formulas that exist in many simple power calculations. And usually there are few different versions of these, of the approximate formula that will depend on the precise assumption that we wish to make. But all of these should be given broadly similar answer, therefore choosing whichever one will depend, of course, on our question of the study. And usually using reliable statistical software is the best option, such as R and SPSS, which we have discussed. And since this will tend to give the most accurate answers rather than simple hand calculation. So for the sample size, the main idea behind the sample size calculation is to have a high chance of detecting a statistically significant or worthwhile effect if it actually exists, and therefore to be reasonably sure that no such benefit will exist if it is not found in the trial. And the number of patients that are necessary to be recruited to a particular trial will depend on several factors. These will include the anticipated clinical differences between the treatments and the level of stat significance that is deemed appropriate for this type of study, in addition to the chance of detecting the difference if it actually exists. So the greater the power of the study, then the more sure we can be, but the greater power remains more crucial to the larger sample size. So a sample size which is too small can lead to an underpowered study and therefore should be avoided. But if the sample size that is required is too large to be reasonable, then the study will not be able to take place because of the costs that are too much or even the inability to recruit large number of participants. So in this case, there has been an appropriate balance between the power of the study and the stat significance which need to be achieved. And there are different methods for calculating the sample size of quantitative studies. This will include also subcategories of different sampling methods. And the first one we will be discussing is the sample size calculation for continuous data. So based on the standard difference, the ratio of the difference of interest to the standard deviation of the observation, then the smaller the ratio of the larger the required sample size of the trial will be. And there are 4 quantities that need to be specified in this case. This will include the standard deviation of the variable, the clinically relevant differences, the significance level, and also the power of the study. So all of these we need to consider or take into consideration when we are designing a clinical trial. So the standard difference is simply calculated as the ratio of the difference of interest to the standard deviation of the sample. And using appropriate nomograms of the sample size that can be extended for specific level of significance and power is recommended to be used in this type of sample sizes. An example of this is the milk supplement study. Let's suppose that we want to investigate whether a daily milk supplement given for a year to 5-year-old children will lead to an increased gain in height. So we know that at this age, children will grow on an average of 6 centimeters in a year with a standard deviation of about 2 centimeters. And then we will suppose that the height gain of about 1 centimeter was considered important for each children. Therefore, to calculate the standard difference, this will be 1 divided by 2, which will equal 0.5. So from the normogram for a trial with an about 80% power and significant level of 5%, then a total of 120 participants are needed. And if we want to have a power of 90%, then about 160 participants will be needed. So depending on the power of the study, the sample size will be calculated. And this is the equation for comparing the two mean values of any set of data. The delta in this case is the expected difference between the two means, and the O, or the underlying standard deviation of the individual value from which the mean would be calculated. And for the Z, it is the chosen one to represent the power that we required, and the second one represent the cutoff point that we need. So the typical value of the first Z is about 80% power, or 1.2 for 90% power. And for the second one, it is a fixed value of 1.96. So let's suppose that under the null hypothesis of no effect, for example, in a group of patients given a placebo, then we would expect to see a mean outcome of about 50 units. Then we would suppose that the standard deviation is 10 units under the alternative hypothesis of using a two-tailed p-value. And in the previous equation, we will replace our values for the previous equation, or we can immediately use our program, as we mentioned yesterday, to analyze the result by entering these data. So if we were to enter these data and get the code, then we will have the following result, in which the mean value will be equal to 22, as you can see here, and this will be the standard deviation of this sample size calculation. So this was the calculation for quantitative data. And for the case of a categorical data, where there is binary outcome, for example, the participant response, such as yes and no questions, the calculation of the sample size is based upon the comparing of the proportions. Therefore, the expected proportion for each group need to be estimated as B1 and B2, with the standardized difference that can be calculated using the following formula. That is minusing B1 from B2 and dividing the result by the mean of the two groups. So another example for the categorical data is a case study of research in stopping smoking or smoking cessation. So let's suppose that we want to compare a new nicotine chewing gum with a conventional device and support. And in the advice group, for example, 15% of smokers will remain as non-smokers at 6 months. Then we would be even interested in improving this result to 30% in the chewing gum group. In this case, the standard deviation or the standard difference will be minusing the two means from each other, as we mentioned previously in this fixed formula. So with about a power of 80% off, with at least 5% of the level, then we would need 280 smokers to have a statistically significant result in a sample size. And if we wanted to compare the two proportions, we will just need to replace our values in this pre-specified formula to calculate the mean value of these two observations. So if we were to enter these data in our program, then we will immediately get the result of about 400 participants to have an equivalent sample size. And in the case of survival data, which are data containing the time to the event component, this needs to be analyzed using survival methods. Therefore, in planning a trial with survival data, the estimates of survival probability at a given time endpoint are required for the power of the study. So an example of this method of sample size calculation is a study in the primary resistance Hodgkin lymphoma disease. So let's suppose that we want to compare the AST with the conventional chemotherapy. Then the conventional chemotherapy has about 5 years survival probability of about 35%. Then we would postulate this with the survival probability that may need to be 50% at an increase of 15%. Therefore, to know how many patients we will be requiring if we want to design a trial with 80% power to detect any statistical significance at 5% level, then from the statistical tables for the design of the clinical trial, that is the mission and compatible result, then we will have a pre-specified sample size number. Where if we want to have a higher power of about 95%, then this would mean that the sample size needs to be increased. This can be reaching to 200 sample calculation. So this is the prefix formula that we should be using in survival curves data to compare any sample size calculation. But we should know that this formula can give the total number of required events. For example, if the end value that we want to assess is death, this will need to be converted to a total number of subjects that we need to take into account, planning the study duration, the length of follow-up, and also the underlying event rate or the disease outcome. So let's suppose that under the null hypothesis, there is no effect, for example, of a group in patients given a placebo, and we would expect to see about at least 50% of patient survival over 5 years. So under the alternative hypothesis, for this example, we would expect about 60% of patient survival at 5 years. And using about 80% power calculation, this will result in about 350 participants needed to be recruited. And if we were to enter these data in our program, then we would be having the same results, but larger in explanation and more data. So this calculation can tell us that the expected overall proportion of death over 10 years is about 0.70. And therefore, 350 deaths given as the estimated from the earlier approximate calculation, and a total of required participants would be about 500. So what are other calculation or considerations for sample size? As there is a strong possibility that not all patients can be recruited to a trial, this will be illegal for analysis for non-compliance, patient loss due to follow-up, and any adverse reaction during the course of the trial. So this will result in a fewer patients available for analysis, and therefore to maintain the appropriate power of the study with extra patients that need to be recruited to ensure that we will have the desired result at the end of the study. Of course, there are some ethical issues regarding sample size, as it is it even ethical or legal to randomize patients to the treatment? And is it ethical not to randomize when there is a promising new treatment available? but its efficacy is still unknown. And also, can a truly informed consent ever be obtained from the patient? And also, when it is legal to use placebo. So all of these are ethical concerns that we need to take into consideration when we are choosing the sample size of any type of study. So the sample size and the power are critical components of the study design, and studies with inadequate power are considered waste of research resources and also arguably unethical when the subjects are exposed to potentially harmful or even inferior experimental conditions. Also, studies with inadequate power not only risk the failure to detect a genuine effect, but they can also result in the majority of parent findings being spurious. That is a particular true in the omic type of studies, while other studies we can look for the possible association between the outcome and the large number of possibly related predictive and even prognostic factors. So if your experimental sample size is fixed and pre-specified, it is still appropriate to input the figures to a power calculation so that you are at least aware of the level of power that our trial will have. So it is good practice to calculate a variety of sample size estimates based on a variety of different assumptions about our experimental outcome. This will help us to illustrate how sensitive the required numbers are to change the assumption or the assumed response. And in the early exploratory studies, when we have no information available to support our formal power calculation, a good rule of thumb, which has some statistical significance, can be used. So the rule of thumb includes a sample size of 12 per group as an early estimation for the sample size. In addition, if the sample size is critical issue, then little relevant information is available to help make the assumption that underlies with a sample size calculation. Therefore, we can try to gather the internal data and conduct an analysis of the data after recruitment of a small number of patients. And we can also use these data to re-estimate the required sample size for the study, and it can require careful planning in order for the study to remain valid. So these can include issues of unbinding or a potential bias that can arise in this step. And as a final step in any sample size estimation or assumptions, it is good practice to think about issues such as patient dropout and also missing data, which can also inflate the estimated numbers to cover the data that are lost for these and other reasons. Also, we should think about the size of the subgroup of the patient within the study and if the subgroup analyses are planned or not. In addition, an important thing to consider is that we need to report our sample size calculations when we are publishing the study. Therefore, we should give full details of the assumption that we went into the calculation, and also the same figures for the estimated form that we have conducted from our calculated data. So, sample size and power are critical aspects of the study design, and low-powered studies are arguably unethical and, in some situations, can result in a majority of apparent finding being wrong. In addition to this, the power calculations are based on the statistical distribution theory, in specific on the overlap between the sampling distribution of a relevant effect measure under the null and the alternative hypothesis. Also, approximate formula do exist for many situations, and many of the variation on these formula do exist with web-based calculations that are available. Also, using reliable stat software can give more accurate results, and all sample size and power calculation should be considered to be approximations of the actual effect. We will now continue with discussing the qualitative research, study design, and method. So, as we have discussed earlier and yesterday, quantitative research means focusing more on the numbers and on the quantity of the participant, where qualitative from the name will have a quality more in the number of the patient. And this type of studies, of the qualitative studies, will almost always have public involvement in the process and will focus more on the opinions of the public rather on gathering general information, because as we mentioned, it will focus on the quality of the gathered data rather on the quantity. So in this type of studies, we will always find public involvement. So why do they include the public in this type of research? This is due to the ethical right, the democratic right with the utilitarianism argument of the study, and it is mandatory for some organizations, and it can help in avoiding waste research because, as we mentioned, it doesn't rely on the numbers of the participants, but rather on the quality of the gathered information. So public involvement for all researchers can be planned before we start our research. So this can be occurring at any point of the research cycle, including the identifying and prioritizing phase. This will have the patient or stakeholder identifying any relevant research topic through the consultation, or it can be in the designing and managing period. This can be conducted to ensure that the protocol and the methods are patient appropriate. Also, it can assist with the recruitment and retention of the strategies, in addition to defining the outcome measures, assisting to steer the project, or even developing the research tool with reviewing any informed consent or personal information sheet. Where in the funding and the commissioning phase, public involvement can be used to review the draft research funding proposal and ensuring the research methods are even ethical, and it can be named as co-application in this phase. While in the undertaking and analysis phase, they can be included to collaborate in data collections, such as with phone apps or conducting interviews and survey, in addition to analyzing or interpreting the data and the result. In the dissemination phase, they can be included to give advice on the venues, for example, for the disseminations, or jointly even presenting the result or contributing to publication. While in the implementation phase, they can be included to increase the likelihood of the result being actually implemented after the end of the study due to patient support and lobbying of the participants. Also in the assessment of the value of the work that we have carried and analyzing the benefit and the risk. While in the final phase, which is the evaluating of the impact, they can be included to collaborate with the researcher to evaluate its impact and even its processes, and the patient public reflects on their role and what they learned from being included in the research. So with the qualitative research, they can be included the methods that are being used or the kind of data produced, but they more significantly focus on what question does it seek to answer about a phenomena, focusing not on how many number we have, but rather on how many valuable information that we will be able to gather. So it focuses on how do we understand the world around us, more like philosophical answers. So there are different ways of knowing. This can be as a positivism method or phenomena. These will include the stable reality, the observable, and the value-free aligned with the nature of the science. There is also the interpretivism, which is not a search for the reality of the world, but rather on how people interpret the world around them. So if we understand what the assumptions are made at the planning stage of the research, we will be able to come up with the best method for studying our question. So why do we always choose, in most of the cases, quantitative rather than qualitative? So according to the labs versus the social research, there is always interpretation to the data. So it is not like there is an absolute answer to every set of data, as for instance, when we get bands or gel, for example, or get some kind of numbers, it has to do with what we set up with the machine rather than what we take as a background and how many we can control. So a qualitative research is a set of interpretive material practice that can make the world visible. And these practices can transform the world, and they turn the world into a series of representations, including field notes, interviews, conversations, photographs, recording, and memes to the self. Also, for the qualitative researchers study things in their natural setting, with even attempting to make sense of our interpreted phenomena in terms of meanings people bring to them. So the key features of the qualitative approach focus on, for example, research question as focusing on how and why rather on how many. In the data collection, it will focus on close contact between the researcher and the participants. Also, it can be flexible and responsive to the context of the accounts. And in regard to the data that are being obtained, this will be rich in details, detailed and extensive in the content, but there are fewer cases than the quantitative. For the analysis, it can help us or allow us for the emergent concept, and it can involve the description, the classification, the types, and the explanations. And in regard to the output, it will interpret the social meaning and represent the social world of the participants. So when can we use qualitative research? This can be conducted, for example, in HPV vaccination, as we needed quantitative studies to estimate the size of the target population, the scale of vaccine production, the service framework for providing maximum coverage, and we will need, for example, in this case, qualitative studies to find out what the parents or adolescents think about this type of vaccination and why do they believe what they do, in addition to how to overcome these challenges in order to ensure that everyone receives this healthy vaccinations. An example of this is the VOICE trial. This was the vaginal and oral intervention to control epidemics in Africa. So a multi-site HIV pre-exposure prophylaxis trial that targeted young women in South Africa and Zimbabwe and Uganda. They had a daily application of 1% of tenoviral vaginal gel plus a daily oral tenoviral and imitriptyline. So in this study, the results showed that there is no effectiveness for the three products that were tested among 5,000 women in 15 different sites. So widespread product of none used the plasma level despite the high end of the study retention rate and also the high self-reported product use, but the result says otherwise. So in order to have more insight into the problem in this case, they conducted a qualitative research to understand what caused this type of problem. So in the qualitative exploratory study, it was implemented in 2010 with the concurrently VOICE trial. It included only 100 participants compared to the 5,000 in the previous trial, and they included focus group discussion, in-depth interview at the study clinic, and even ethnographic interviews. So according to this study, what they found out that HIV stigma resulted in the non-use of the gel they gave them and the vaccine. They also had fear of being labeled as having HIV, therefore they didn't use it. And they have ambivalence about the research process and were confused about the use of the treatment to prevent infection, which needed to balance the trial participant with other priorities and social relationships. So from this example, you can see the difference between a qualitative and quantitative research. And therefore, we can assess that qualitative research in most of the cases will be needed to assess its impact. So the key differences in the method and purposes of quantitative versus qualitative research. In the quantitative, it will have more numbers, while the qualitative will have more words. In the quantitative, they will test the hypothesis, while in the qualitative, they will produce the theory. And many other differences, including in the number of the participants they are being recruited, the type of questions that will be asked, and even in the statistical analysis of the gathered data. In addition, it will have differences in the contextual content, as the quantitative research will extend on existing form of homelessness, for example, but it will have more characteristic of those people. Where in the qualitative, it will reflect on the nature of different forms of available homelessness in this case, and it will have more on the experience. It will also have different explanatory and evaluatory methods with generative data that is being different in regard to, for example, predicting any future levels of the issue or even suggesting strategies for improving the issue in the future. So what about the different sampling technique in the qualitative methods? In the qualitative, the sample aim will be in-depth exploration and data in context, while in the quantitative, it will need to measure the extent with statistical correlations. So in the qualitative, the sample recruitment will have map of diversity, including key group and dimensions, and it will most importantly have a smaller sample size, where in the quantitative, it will have a larger sample size. So the sample type can be purposive, meaning it will have deliberated selection on the basis of knowing their characteristic, while in the quantitative, it must be a random probability or else it will result in bias of the sample. So the sampling in qualitative research will focus on four techniques. The first is purposive, theoretical, convenience, or snowballing method. All of these will depend, of course, on our questions that we need to answer. So in the first type, the purposive sampling technique, this is accreditation-based sampling. It is selected with the purpose to represent a type in relation to the key criteria that is suspected to be relevant. This will include the socio-demographic, the experience, the behavior, depending, of course, on the issue that we need to assess. It will also have two aims to ensure that all relevant criteria are represented. It will also include diversity within each key criteria. Also, there are different approaches to this method, including a homogeneous sample, a heterogeneous sample, or extreme cases with key informats. Where in the case of the theoretical sampling, this is a sample based on their potential to contribute to the development and also testing of theoretical construct. Associated with the development of a grounded theory with initial sample selected, data analyzed, further sample selected to refine the emerging categories and theory, and also it can be useful in exploratory research where little is known about a particular phenomenon. In the snowballing and the convenience sampling, the first one, it will include a series of introductions to a new sample from the research participant. It will be useful for hard to reach area or easy to ignore groups. Where in the convenience sampling, we will just choose the sample according to the ease of access. But this will, of course, lack a clear sampling strategy and may not be as reliable. So how can we inquire about these qualitative studies? This can be done according to observations as it can be combined with the participation, also according to archives or other documentary sources, including the media, or it can be conducted through interviews. These interviews can be semi-structured, a narrative interview, a one-to-one interview, or even through focus groups. There are, of course, different types of interviews. These can be structured interview, which are mostly used in quantitative survey. This will include a set of standardized questions with fixed wording and order of questions. This will also be predetermined categories and as a part of the large sample surveys. Where in the semi-structured interview, this will be mostly in qualitative studies, and it will be more flexible with standardized interview schedule or with topic guide even, and it will include exploration of the emergent themes and ideas. But in this case, it will have limited recognition or disclosure. Where in the case of an unstructured interview, this will be also in qualitative studies. The interview style will be unstructured, meaning it will have open questions and more freely asking with some gentle guidance, and it should be including biographical stories with a narrative included. So in this type of interviews, we must have a topic guide. This will be a flexible guide that documents the scope and the coverage of the interview. It will have key sections with key subtopics, and it will also have ordering of the topics according to their discussion. And it should begin with easier and less sensitive questions with a positive end, and it should have pilot interview procedures and topic guide development in a pilot study before mass implementation. So the questions for depth and breadth should be included. For example, depending, of course, on the type of the questions, discussing the short versus the open questions, such as yes and no questions, and they should avoid any leading questions or double questions, which can interrupt the participant. Also, the content mapping to raise any issues or to explore any issues. So, to conduct any interview, we must have rapport and trust between the interviewers and the participant. Also, it should be natural and reassuring approaches to the participant, and we should avoid self-disclosure as we should leave the patient or the participant talk more freely. In addition to this, we should listen for them carefully and remember that our role as a researcher, not as a counselor or advisor. Also for the audio record and to have matching interviewers and interviewee, depending, of course, on the likelihood of similarity between them. So one of the types of interviews are focus groups. These should include about 6 to 8 participants, and the interaction between the participants should be allowed with compositions that is carefully selected to encourage the exchange of views and experience, depending, of course, on that issue that we need to discuss or analyze. So there are some main differences between the in-depth interviews and the focus groups. In the in-depth interviews, it will have more personal stories with even detailed or complex issues, and it could include more sensitive issues or questions. Where in the focus group, this will be generated by the group interaction, and it can have broader understanding of issues and shouldn't include sensitive information or sensitive questions. So after we have gathered the data for the qualitative research, we need to consider the best available analysis method. So we aim to understand the phenomena being studied, and we should go beyond reporting, but toward rather interpreting of the information that we have collected. And we should identify the information and the themes, and should be also reflexive of what we observe. So how can we manage the qualitative data? The data security is important as qualitative data can include personal opinions and personal details. It should be coded with ID and fake names for the patient used with stored data and transcripts. We should also protect the confidentiality and the anonymity of the informations which were reporting result. And in this type of studies, we should start the analysis as early as possible as we develop the topic guide and conducted our first interview. As this will be a iterative process, meaning that we will have new information from the initial data collection, and subsequently we can incorporate it into the topic guidance and also the sampling. So this can be useful of taking the notes during the data collection process and the field dairy in general. So the framework for the analysis process include the data familiarization as reading of the interviewer transcript, listening to audio recording, and even using field notes. Also creating the thematic framework by developing a coding scheme of the themes and indexing or coding with signposting the interesting bytes, not rather than the meaning. Also using chart by creating thematic chart, providing a picture of the data, and allowing a visual assessment across the individual cases and themes. It should also help in the mapping and interpretation with exploring the associations between the themes and developing conceptual understanding. This will also help in developing categories, capturing its essential meaning, and going even beyond the descriptive information. So the approaches for the framework is to allow within and between cases analysis, to also increase the transparency of the data analysis process, and to allow the existing theories and ideas about the area under study to be taken into account within our analysis. Also, analysis is both inductive and deductive, with some themes as a priori where other are emergent from the data, plus any new analytical themes. There are, of course, other forms of data analysis, including the grounded theory analysis, which will help in the generation of analytical categories and the identification of relationships between them. Also, it can be conducted in the data collection and conceptualization of the continuous until the categories and the relationships are saturated to the new data added. Also, there is the content analysis, which account for the numbers of times that something is being mentioned in the field notes or even in the transcript as a rough estimate for frequency. Also, the hermeneutical analysis, this is not looking for the objective meaning of a text, but rather the meaning of a text for the people in the current situation. And in the discourse analysis, this is a linguistic analysis of an ongoing flow of communication, and it can help in finding patterns of questions and who dominated the time and how much with other patterns of interaction. And for the phenomenology, this will include how individuals experience the world with emphasis on the ideosymmetric meaning to the individuals and also not having a shared construction as such with the hermeneutical analysis. And in the last type, the narrative analysis, this will help in exploring the participants' stories as what they choose to share about themselves with the interviewer. So there are three final concerns in a qualitative research. This will focus on the quality, the validity, and of course, the generalizability of the gathered data. So the quality of the qualitative research is a part of a much larger epistemological debate about the nature of the knowledge that are produced by the qualitative research. So how do we judge the quality of a qualitative research? There are, of course, some standard guidelines that need to be followed. So these will include the consolidated criteria for reporting of qualitative research. This is a checklist of about 32 items that we need to follow. There is also the relevance, the appropriateness, and the transparency of the soundness of interpretation in general. And for the validity of the qualitative research, this should be a transparent and comprehensive information. It should also provide a context for the research and give a clear evidence and interpretations, including the analysis of deviant cases. Also, the triangulation of to provide the overall interpretation with multiple methods used or even data sources with previous research studies. But we also should be reflexive of the informed data as the researcher role is affecting the research process with the outcome and the interpretation. And even in the how the data is being collected with the personal biases or any assumption or even the personal characteristic. So the third concern in qualitative research is the data generalizability. So the generalizability of the qualitative research focus on three kinds. The representational, whether what we found in a research of a sample can be generalized or even held to be equally true to the parent population from which the sample is drawn. Or if it is inferential, whether the finding from the particular study can be generalized or not, or even inferred to other setting or context. And it should be theoretical, which should draw on the theoretical prepositions of the principles or statement from the finding of the study for a more general application. So we will be taking a break now for Salat al-Maghrib, and then we will continue with the implementation of the research project. Assalamu alaikum wa rahmatullah. Welcome back, everyone, in our guide to health research webinar. So, as we discussed previously, the type of qualitative research with the critical appraisal, we will now continue with the implementation of a research project. So, we have learned all of the necessary steps to undertake a research. Now we will start off the implementation process. So there are some techniques or methods for the data collection. These will depend, of course, on the type of data that we need to collect and could be based upon observational data collection. And this will consist of observing a phenomena in order to gain insight into how and why things happen. And it can include direct and indirect observations by a human observer of a certain phenomenon. This will be done in order to be able to describe it and also to help in contribution to its understanding. So it is also based on the real event, as it is actual behavior that the observed, not the verbal response as in the questionnaires. And also it has a major advantage of observations of its directness. Also, in the observations, we don't speak to the participant about their views, but rather we watch what they do and also listen to what they say. So it is merely based upon observations. So it is rather a labor-intensive method because we will need a large number of participants and of researchers to conduct the observations. And the processing and interpretation of these acquired material is difficult and also considered time-consuming because we are observing rather than collecting specific set of information. Also, the observation is used especially in the descriptive research where there are many open questions, and it can also be used in the experimental research when the researcher has a specific expectation about the result they are seeking to obtain or acquire. In addition to this, it is important to define precisely the things which we need to be observed and use an observation schemes which should be reliable and also structured, such as a system of categories when one can also record this result. It will also help in establishing the observation, which is the most suitable method of data collection, and can help with working in several observations recording of the same behavior and using audio-visual recording equipment so as to find the pattern of the behavior. In addition, it will help in ensuring that the observed are unaware of being observed rather than having a fear of affecting their behavior in a certain manner. And it can also help in carrying out a pilot study to check whether the method worked as expected or not. So, in general, observations method can also include reviewing the record, observing the sign, and even measuring the heart with the checking the temperature, for example, or blood pressure, any type of making observation rather than assessing after an intervention. The second technique or method for data collection is the questioning or interviewing. So factors are relatively easy to get at, even though that errors can occur due to lapses in the memory or even to the response biases of various kinds, such as age may be claimed to be less than it is by the middle-aged people. Also, the disadvantage of this method is the possibility that it can make the researcher have direction with the subject towards certain answer. This can be an intentional bias in most cases. So in addition to this, the perception of the responses in assessing the questionnaire with the foreknowledge of the aim of the researcher and also answering the desirable question or answer based upon the facial expressions in the interview. And the interviews are kind of a conversation with a purpose or a topic guide. Also, it is initiated by the interviewer for the specific purpose of obtaining the research relevant information and also on focusing on the research objectives. So in these cases, the interviews are flexible and even adaptive to a way of finding things out, where in the case of a face-to-face interviews, these can offer the possibility of modifying one line of inquiry, and also interviews can be conducted over the phone for ease of conduction or for the participants. And also the interviewing is time consuming at most cases, and any interview that is less than half an hour is unlikely to be valuable. And any other type of interviews going over an hour can be unreasonable demand of the participants or making it very difficult to even recruit participants. So asking can also include conducting focus group discussion, administering a written questionnaire, nominal group technique, and any other technique such as case study. It is also important to know that data collection could be collected using more than one technique, depending, of course, on the question that we seek to answer. And of course, there are some different tools for the data collection, and these can be applied during the collection process. And a particular technique can even involve different tools depending, of course, on the questions that we seek to answer or the type of the data that we are actually collecting. So the tools can include, for example, weighting scale, measuring tapes, or microscope with observational checklist, form questionnaires, etc. Any tools meaning that can help us in answering the question which we seek to answer. So in the questionnaires and the forms are used largely to record the factual information such as the subject age, the blood pressure, or the treatment group. And these are commonly used in clinical trials to follow a patient's progress and are often completed by the responsible investigator in general. So the principles of the questionnaire design should be followed when we are conducting a questionnaire-based method. And this can include the content of the questionnaire, as it should be designed to ask the minimum question possible to collect the required data, with each question justified in terms of objectives of the study and according to the types of the available questions. These can be opened, closed, or enclosed questions. So in the open type of questions, these can allow the respondent to answer in their own terms and are used for the non-categorical variables, such as the age, the date of birth, etc. So they can even take more time and effort to complete because these are standardized set of answers that need to be followed. And in the closed questions, these can reduce the possibility of the interviewer interpretation and also the coding bias, which can facilitate the processing and can simply be answered by circling or tickling the response. Also, in the closed questions, where only one out of the selection of the response is allowed, this can be, for example, by ticking a box, then the final option should always be if others, please justify. So we will be just taking a quick break for 5 minutes and then we'll continue. Assalamu alaikum wa rahmatullah. Welcome back, everyone. So, as we were discussing the type of the questionnaire designs, and we have discussed the open and the closed questions. So, in the closed questions, it can also include the Likert rating scale, such as doctors, for example, can strongly agree, disagree, or etc. Also, the visual analog score, or the pain as bad as it can be as no pain. So basically having a specified score. In addition to this, we should pay attention to the wording of the questions, as it must be written in simple, non-clinical or technical languages, and the wording should be avoiding any suggesting of a particular answer is preferred to avoid any bias. So, also, the questionnaire layout should have an introduction and explanatory notes and also instructions with example on how to complete a particular questions that should be brief. Also, the method of administration should be included, as the questionnaires can either be self-administered, interviewer-based, or computer-administered. So each type has its own advantages and disadvantages, with the computer one being the best and need less time for administration and more easily administered. Also, we should have methods of administration, such as the questionnaires, as we mentioned, can be computer-based, as it will need less time with the question sequencing. That the question should follow a logical sequence, following as far as possible the sequence which the respondent may actually expect to follow. And it should proceed from the general to the particular question, so with the less sensitive to the more sensitive questions. And when a response to a general questions, make further response on the subject of any irrelevant or unnecessary questions, then these can be introduced and should be as simple as possible. So in addition to this, the recording of the response can be included, as the objective is to obtain a clear answer to each question. And it is important to ensure that when we are using closed questions, all the possible responses are included in the list of options. And failure to do so will even result in biased response, thereby reducing the care in the completion and in the corporation. And for coding, it is important from the beginning to decide on the coding scheme to be used for each variable. And this will help to ensure that the appropriate variables are being collected for the analysis. And using a coding guide should be constructed, which provide the key to the meaning of the response. So in addition to this, there are some general considerations, as questionnaires should be subject to the pre-testing and the piloting. Also, the assessment of the validity of the questionnaires should be conducted from the beginning to ensure that we are following the correct guidelines or the correct procedures. Also, it is desirable to use the standard questionnaires, as if, for example, they will usually have been used and proved satisfactory previously in previous research, and it may have been validated or will even facilitate the comparison of results from our study with the data collected by any other workers. In addition to this, we should always plan for the data presentation and the analysis, as after the selection of the study variable, the design, the sampling method, and data collection techniques, a plan should be put in for the data collection. And this plan should include the type and the number of data collection tools needed, such as questionnaires or weighting scales. Also, it should include the categories and the numbers of data collection personnel, such as dietetic technician, for example, or physician, depending, of course, on the type of the study. Also, the duration and the content of training that is needed, and the schedule or the timetable for data collection. These can include starting date, preparation of the tools, and even time of the study phases. Also, the logistical support, such as vehicle, food, and drink transport, any other included thing in the study which need to be cared for. Also, estimation of the budget, such as the budget tools for training or for photocopies, etc. In addition to the plan for processing and analysis of the data that must be prepared before it is collected in the field so that it is still possible to make any necessary changes and even anticipate the future events. Also, this is necessary so as to ensure that all the needed information is going to be collected in a systematic and even in a standardized way. And it will also avoid the collection of any unnecessary data or over collection in general. So the content of the research plan should also include sorting the data or the data collection by making careful comparison between the copies of the collection sheet and learning the differences between them, such as putting the likes with the like, and if there are more than one questionnaires, putting each one separately, or even the cases with the control, for example. Also, performing quality control checks, such as data cleaning and checking for any missing questions, any range check, or interviewers that must be trained to follow any standard procedure protocol. And also the data processing, as it could be either manually or computerized, and both of which can involve coding, which is the translation of data into symbols of appropriateness for the analysis. So this is actually true for the cases of analysis in the qualitative data in specific. Also, it should include the preparation of tables or any other analysis that will be needed. In addition to this, the decisions on the sequence in which the table should be analyzed should be included in the plan, with estimating the total number or the total time of analysis, and the decision on the staff needed for analysis, and estimating the total cost of the analysis in general. So how do we format the dissertation of a proposal? The title should be sufficiently detailed to inform the reader what the researcher or the student is proposed to do, with a maximum of 2 lines. And it should include a researchable problem with specified population, place, and date. And also, it should be in the format of a positive sentence, for example, questions that are not universally accepted. Afterward come the introduction, which should include selective review of the background to be put in the dissertation research into the context of any appropriate recent relevant literature. This can include about information about other work in the field. And it should include the key research report, the reviews article rather than the textbook, in about 2 to 3 pages, with outlining the problem, issues, or topic for the dissertation, and also for the need to taking the research at this particular area. So after writing the introductions comes the research question. This is a statement of the purpose of the proposed research in the form of an objective that should be stated in this part. And this can also be the hypothesis, which is not included in the design, but it should also include the objective study. So after writing the research hypothesis and objectives, we should include the methodology and also the design issues or the study base, such as the facility or the community clinics which we are using. After that comes the study design in regard to the time direction. Is it a retrospective study or a prospective study with the data gathering method or methodology? And in addition to the sampling technique or any other required data to be collected that need to be cited in specific. So these can also include the techniques, the tool and equipment with the resources, and then adding the references with the timetable of the research. So the most important thing is the ethical issue that should be considered carefully. And one of these issues should be the permission to take the study, and is often obtained from the manager of the place where the study will be carried out, such as in the hospital, in a nursing home, or a nursery, depending, of course, on our study. So the second ethical concern is the consent form that need to be obtained from any study participant. And in general, in the methodology section, it should be between 1,000 to 1,500 words. And in the final word, the final chapter, this should be the dissertation and the presentation or discussing with a total number between 10,000 to 12,000 words with any acknowledgement and legends. So after discussing the quantitative and the qualitative studies, we will now continue discussing the mixed method research studies and any other type of study that we can encounter. So in clinical practice and research, these can be conducted to assist the diagnosis of a disease, to treat a disease, or even to prevent one. So depending, of course, on our aim, this will decide the type of the study. So what is a clinical research? A clinical research is done to improve any of the above. These include the diagnosis, the treatment, or the prevention of a disease. So any research conducted to improve those mentioned points will be regarded as clinical research. So we shouldn't fixate on the definitions, as the clinical research can be research to improve the clinical practice, where the experimental medicine is an experiment where the human is the subject in this case. And it can include also translational research, which is bench to bedside, and hack, which include mostly laboratory-based type of research. So how can we learn about human disease from the beginning? Before we can start any clinical trial, we can start our research process in the in vitro studies. These include conducting a research in a cells. So it is referred to a cell in a dish. And in this case, we can manipulate the cells by having the desired impact we want on them. And the disadvantage in this type of studies is that in vivo environment is lost in most cases, and treating a cell is a long way from treating a patient, but rather considered as a simple exploration of these effects. And where for the animal model studies, this is another way we can learn about human disease. These can, for example, be conducted for assessing cancer studies, in which the advantage can be that we can sacrifice the animal and solely look at the tissue to see any changes or to detect any change in the tissue. But the disadvantage is that animals will, of course, have different effects or human bodily effects than the humans. And these animal models may partially resemble the human disease, but even if we did manipulate the genes of the animal to look like the human genes, it will not be 100% the same. Also, another method that we can learn about human disease is using dead bites of human in, for example, tissue banks or biopsy, pathophysiology, as it can have the advantage of assessing the exact tissue of the correct animal, and it is the correct disease in the correct environment. In addition to this, the disadvantage that we can only assess the effect on a certain time point, which is normally when the patient is dead, and we cannot apply any intervention in this case as the patient, as we mentioned, has deceased. Also, another type, or the most common type, which is clinical trial in living participants. These are the basis for experimental medicines and clinical trials, and it can help in studying the true disease using the correct environment, and the intervention is actually possible in this case. But these are very limited in what we can measure and often are very expensive in terms of the tools that are required and the number of participants that need to be recruited and these types of limitations. Also, in the epidemiology studies, these can help in identifying the gene responsible for certain intervention or certain impact on the body. But it can also have clues only, as we cannot actually detect the true effect in large populations because we cannot apply a large population site intervention. So, as we mentioned briefly yesterday, the drug development stages take very long periods and can last between 10 to even 15 periods to approve any certain medication, according, of course, to their feasibility. So, why do most cases or clinical trials actually fail to meet the final guidelines? These can be due to safety concerns, efficacy concerns, or even due to strategy issues with the planning of the research. So, as we can see, the most commonly cause for the rejection of any clinical trial medication is due to missing efficacy of this medication. Also, it can be due to regarding safety issues, mostly between the years of 2013 and 15. This was the reason for rejection of a large number of medication. So only about 50% of clinical trial will survive phase 3. This is in regard to the medication trials. So how can we improve the decision making about which drug to develop? This will be a complex question to answer, but everyone agrees that we will need a better translational medicine, better clinical research, experimental medicine, or what we even would call not relying entirely on mice in the preclinical trial phase. So there are some tools for experimental medicines. These can include biobanks, which include the human tissue repositories, and also the imaging facilities with high-tech and novel imaging, such as PET scan and MRI. Also in the genetic with the molecular pathology laboratory labs capable of high throughput sequencing for genes, and also in the clinical research facilities with dedicated facilities for conducting human research. So in biobanks, many diseases have the expression of the drug target but need to be validated. And these can include tissues, not just blood, and it will need rigorous initiation, linkage to the clinical data with appropriate governments to be implemented ethically and correctly. So an example of this is a post-mortem brain from a patient with multiple sclerosis. And in this case, we can see a radio gland with the blinding of the macrophage protein. So while in the imaging, these can be helpful in detecting the pharmacokinetics and can help in detecting and quantifying the drug itself or the drug target. This will help us also in answering the questions, does my drug get into the brain, for example, and what the dose do I need, and also how long does it take to last in there, meaning in the excretion and in the time for action. Also, in regard to the pharmacodynamic, it can help in detecting or even in the quantifying of the marker of the drug effect by asking ourselves if the drug actually does anything. And it can be helpful in trials for chronic disease, which are even long, large, and therefore expensive. It would be nice to have an indication of the drug action and the molecular changes that precede the structural changes, which actually can precede any clinical change. So it can be helpful at every stage of the research process. So example of this is using a PET scan in the receptor of occupancy studies, as it can show the expression for certain receptor. And if it was in this example repeated after 1 hour, you can see the detected change in the imaging with the appropriate dose curve. So this will depend on the drug doses that we can see in the scan or in the imaging. So this can also be helpful in the MRI assessment of RB structure and function. While a more simple example of this is using the reproduction of the painting of a chromosome in general. So example of this is having 6 participants of both gender with normal BMI and having a crossover double-blinded randomized studies. So the hypothesis for this study is that BYY reduces the hunger and then the food intake. In this simple example, we can assess this by asking the patient, how hungry do you feel right now in different time or hours after consuming their meals? So the effect of BYY can be interpreted based on the patient response, and therefore we can conclude that it, for example, reduced food intake by certain percent and its effect on any other parameters. So how can we initiate the clinical research? We should define the question and design the study by obtaining scientific peer review secure funding and prepare the study document. Also, we should obtain the ethical approval and the management permission for any additional requirement. Therefore, we should always finalize the study document and register our study from the start. So these are a simple planning and funding procedure for conducting a research. So another major point of consideration in research is in specific in any intervention that is applied in the patient is assessing for the absorption and the distribution of the administered medication or diet in general. So in the absorption, this will be different according to the site of intake, if it was an oral drug or even an injection. So there are differences between the two processes which are arbitrary. So we can measure the concentration in the blood of any administered medication, as the blood is the delivery route if the medication was taken in blood. So there are the passive movement of the drug across the membrane and should be accounted for, as in a plasma membrane, the lipid bilayer can encapsulate the cells, where the basement membrane layer of the cells can also intervene with the absorption facility. So an example of this, how propanol can cause nightmare while atenolol doesn't. So the drugs can vary in the water lipid solubility, and this effect can show that they move across the cell layers. And in the lipid soluble, lipophilic often can cross the plasma membrane, while the water soluble drugs often doesn't. So propanol can cross the blood-brain barrier, while atenolol cannot. Therefore, this will explain the effect in the nightmares that develop with this certain medication. So how does the drug move through the cross-cell barrier? This will be through either paracellular pathway or a transcellular pathway. In the paracellular pathway, these will be caused to be done by the tissues in the gaps between the cells, and also will depend upon the drug size and the drug lipid solubility, or if it was hydrophilic. Also, another major consideration is that medication or drugs actually affected by the organs which have the tightest barrier. So the leakiest cell layer ever in the human body is the skin layer, which will be followed by the GI system and then the urinary tract. And after that, the tightest cell layer is actually the blood-brain barrier. And for the transcellular transport, which can feature most importance, these will include the graphs of the permeability that increases as the size decreases, and as it can increase with the lipophilicity that will increase. So the drug size is more important in comparing the blue drug versus the red drug in the cases with the same permeability and the difference in size. Also, there are different forms of transport of medication across the cells. These can include the active transport, which will have only the B glycoprotein for the active transport, and mainly through the gut wall and the brain. There are also different drug responses in the patient with the B glycoprotein mutation, therefore will depend upon the genetic constituent of the patients. Also, the bioavailability of the drug that reaches the systematic circulation is of paramount because it will determine the action or the effect of the drug to be administered. So all drugs have about 100% bioavailability when they are given by IV, and there is rarely 100% bioavailability when a medication is taken orally. So when the medication is taken by mouth, this will lead to destruction by the stomach acid or the bacteria, and it will either be too big for the hydrophilic to be easily crossing the gut wall barrier, or it will be metabolized in the gut wall or efflux transporter. Therefore, any oral drug tend to be lipophilic or even small in size to be absorbed more well. And after leaving the GI, it will lead to the portal vein where it is metabolized in the liver, with after that moving into the systematic circulation. So the percentage of the drug that reaches the systematic circulation defines the bioavailability of the drug. And if, for example, we take 1 gram of paracetamol and only 800 milligram reaches the systematic circulation, then we can say that the bioavailability of this medication is 80%. So how can we measure that? We actually cannot measure it specifically, but rather based on some assumption and the area under the curve that will present the bioavailability of the medication. This will be assessed based on the plasma concentration of the medication and the time that the plasma concentration was assessed. So an example of this is the lactululose versus the augmentin, and this can lead to higher bioavailability in regarding to the IV versus the oral administration. So what causes the variability in the bioavailability? As we mentioned briefly, this can be affected by the stomach pH as highly variable throughout the day, and it can even be increased with food. That's why we say for some medication only to be consumed with food. Also, the exposure to the stomach pH can lead to interdigestive cycle and reduction in the gastric emptying. This will be affected by the food intake, if it was solid or liquid diet, and by any other medication intake, or even the disease status, as for example, in diabetic patients. Also, the CYP enzyme in the gut wall and the liver can be affecting the bioavailability of the medication due to the genetic differences and altered by the food and other drugs. So absorption is highly variable. Does it even matter for these medication? Of course it does, because we want to have a highly observable medication to have a certain effect. So it will depend on where on the curve that we are and this the linear regression that we want to achieve. So an example of this is calcium, which the agonists that will need a low occupancy, where the antagonists will even need higher occupancy during the development phase. This can be due to the fact that they are blocking the endogenous agonist from being observed in general. So the absorption and the distribution can be mainly passive process that is affected by the membrane factors such as the gap between the cells and even the drug factors, including the drug size and if it was lipophilic or hydrophilic. Also, it will be affected by the bioavailability, which can be highly variable during the time of the day, and even the volume of the distribution that provide clues to where the drug has gone, and also it can determine the half-life of the medication. So another major consideration when we are having a clinical trial with medications is the renal elimination time or the clearance from the body of that medication. So we mentioned previously that the renal distal tubules will be the second in the most tightest cell layer ever for the drug to be absorbed. So it will depend on the glomerular filtration rate, the secretion minus the reabsorption, which will be ejected in urine. So the glomerulus is our leakiest membrane, as all drugs that have less than 5 kDa filtered will be, regardless of the charge of their lipophilicity, be excreted. This will be conducted in 125 milli per minute, and it can enter the plasma in about 25 minutes, therefore leading to a full bladder in about 3 minutes after the consumption. And in the tubular reabsorption, the lobes of Henle and the distal tubules will be the tightest cells in the renal, and also the sodium is actively pumped from the filtrate to the plasma, and these are the pumps that the diuretics actually block the mechanism of action for, and therefore the water can follow by omesis process and leading to a more observed impact. So the glomerulus can actually be filtered from the plasma to the filtrate, where the distal tubules will have the very tightest cell layer, even in the paracellular that is not available. Also the lawns of the pump that is back in the transcellular phase, and almost that all water will be reabsorbed in the renal tubules. So the drugs will have a highly concentration in the filtrate, and in regard to the hydrophilic drugs, these are actually trapped and eliminated in the urine, while the lipophilic drugs can escape, therefore be reabsorbed into the blood. So how does this lipophilic type of drug is actually eliminated from the body? These will be eliminated through the liver, or while the hydrophilic drug, as we just explained, will be eliminated through the kidneys. So in renal impairment cases, these can affect the drugs in three ways: by reducing the renal clearance of the drug, or even reducing the renal clearance of the drug metabolites, where any extrarenal effect of the renal impairment will actually affect this clearance pathway of the administered medication. So in the case of uremia, this can reduce the expression level and also impair the function of several metabolic pathways. This can include impairment of the metabolic enzymes, such as in the gut wall or even in the skin. And it can also affect the transporter molecules, including the organic anion transporter, and even with the multi-drug resistant protein, and will eventually result in more drug being absorbed from the gut in specific, and less metabolized in the gut and the liver, and also more entry into the brain, crossing the blood-brain barrier. So how can we quantify renal disease to guide us in the dosing of any administered medication in research? So our first guide is the glomerular filtration rate, but it is only part of the renal elimination, as the elimination will equal the filtration and the secretion minus the reabsorption of the lipophilic drugs. But also we should predict the renal drug handling. And the glomerular filtration rate can be quantified by measuring the creatinine, which is regarded as a natural drug, and it can constantly be produced by the muscles at a fixed rate, and the filtrate of the glomerulus, which is not secreted or even reabsorbed. Therefore, the changes in the creatinine can reflect the change in the filtration rate. And this will, of course, depend upon the gender and the age, and also any other medical conditions. So our options in this case is to reduce the doses or even increase the dose intervals by dividing the dose on multiple injections in the day. So in summary, in the case of renal impairment, for conducting or carrying any research with a drug intervention, we should eliminate the hydrophilic drugs to be eliminated by the kidneys, but the kidney cannot eliminate the lipophilic drugs. But it can also eliminate their side effects or metabolites, and the impairment can reduce the elimination, but also reduce the metabolism of the lipophilic drugs via any indirect effects. But also in the case of impairment, it is relatively easy to quantify and justify for any higher dose elimination. So another major consideration that we need to take into consideration when we are developing a medication for any clinical trial is the half-life and the clearance of the drug. So the plasma half-life is defined as the amount of time required for the plasma drug concentration to be reduced into half. So an example of this, if we have a socket of water, then we will plug it with the kinetics, then the amount of the eliminated proportion to the drug concentration will be appropriate for the dose. So the bucket kinetics is the amount that was eliminated that is consistent and also where the concentration is irrelevant. So if we did eliminate the proportion to the drug concentration, this will be represented in a fixed formula where we can actually calculate the half-life of the drug. So the biological half-life is defined as the amount of time for the biological effect of the drug to be divided into half. So we don't actually formally measure this effect, but rather we would need to consider it because the effect on the clinical trial design in general, specifically in the crossover trial, and we will expect the outcome to be more specifically measured. So the clinical question for this case is if we had a patient taking 5 times their normal atenolol dose, then this patient would feel dizzy and their pulse will be 45 beats in a minute. And if we assume that this patient is admitted to the hospital, when can this patient be actually discharged? We will know by this table that the atenolol half-life or half-time will be 6 hours. Therefore, the time of the discharge for this patient would be at 6 hours after the consumption. While another question can be is that the anesthetic discovered that a spinal surgery patient forgot to omit their aspirin, then we would check the half-time or half-life of the aspirin to ensure that the patient wouldn't face any side effect. And based on the previous table, we would figure out that the aspirin half-life is 2 hours. Therefore, the patient in this case would be cleared. So what are the similarities in the plasma and the biological half-life? These are always similar in all of their three conditions that they met. These include the concentration in the plasma that is related to the concentration at the site of action, and also the drug that binds to the target in a reversible manner, in addition to the effect which rely on the drug actually being at the active site. So in the example of aspirin half-life after 15 minutes will be reduced, where the biological half-life is actually few days. We are more interested in the plasma half-life as it is the major, will have the major effect or impact on the patient condition. So why does the plasma half-life is not equal to the biological half-life? This can be due to the fact that actually the medication will bind in an irreversible method. And the biological half-life is longer than that of the plasma due to the fact that it actually binds to the platelet. So the platelet have no nuclei and will live in the body for 8 days before it is cleared from it. So the plasma half-life doesn't change with any indication because it is specified to the plasma, while the biological half-life may actually be changed in the specific indications. This is due to the fact that due to the platelet inhibition, depending on the patient condition or any other stress factors such as fever or pain. So if we went back to the first question in the example we had of the atenolol half-life, which is 6 hours, then if the patient took 5 times their doses, and then we can actually assess the patient half-life of that medication, which was 6 hours. In this case, the patient can be discharged in the next days because the biological half-life is actually the same of the plasma half-life. While in the case of aspirin, which had a half-life of 2 hours, then the patient cannot have the surgery at 6 p.m. because the biological half-life isn't exactly the same of the plasma half-life. So an example of this is that of how quickly can a lake be cleared, and how do we need to know that according to the size of the lake and the flow rate of the excretion from the water. So the half-life will be proportional in this example to the size of the lake and also the extracted flow rate from that lake. So all of this will, of course, depend upon the patient condition if we were to reflect them on an actual case. So this will be dependent on the volume distribution and the clearance with the half-life of that medication. So the volume of the clearance per minute is expressed as milli per minute and can be for most drugs that are cleared by the liver than those eliminated by the kidney. So we can use this equation or fixed formula for calculating the clearance rate of any medication. Therefore, in summary, the plasma and the biological half-life are not always the same, while the biological half-life can depend more on the system than that of the drug. And also the same medication may have different biological half-life, depending, of course, for each effect on the human body. In addition to this, it is only a relevant concept for the first order of kinetics that need to be implemented, and the determinant for their half-life will be the volume of distribution and the clearance rate. So what if this clearance rate wasn't as we expected? Then this will lead to adverse drug reaction. So any medication, even if they were used correctly, they will have a potential adverse drug reaction. And every prescription will require risk-benefit analysis, as some tactic will include the very rare adverse drug reaction to be used more often, where some will explicitly have a very higher percentage of adverse reactions and should only be used under careful consideration in very controlled clinical trials. So defined as the effect of the drug, which is it a desirable effect or uncomfortable effect? And does it occur with a normal dose or with an overdose? Also, these can include the therapeutic indexes such as bleeding from high warfarin intake with certain medications. So why do we actually consider the adverse drug reactions? As 5% of hospital admissions will suffer from adverse drug reaction, and 15% will involve an actual life-threatening adverse reactions. So who is at risk of developing these reactions are the elderly, especially if they were recruited in certain type of studies, because they will be using multiple medication and will have trials involving the pharmacokinetics. Also, they can be more dangerous in patients with hepatic or renal impairment and in neonate in general, because their body still cannot tolerate any type of medication. So there are 3 top classes of medication that are commonly causing adverse drug reaction. These include warfarin, aspirin, and non-steroidal anti-inflammatory drugs, with also diuretics. Therefore, any type of these medications should be used under careful controlled clinical trial or conditions. So these are actually classified according to their dose and their allergic reaction, or even their the common of the adverse reaction. So when we are developing actually a clinical trial, we would need to ask about the dose reaction. Will it occur on the first dose or on a higher dose? And what would be the symptoms that will develop for those patients? So an example of this is the effect actually common? It will be most common in the case of atenolol, for example. And would it be a predictable side effect? In this case, yes, it would be predictable. and it will most commonly have an allergic reaction, and also it will be dependent upon the dose that we are administering. So when we are having a research clinical trial with medication, we need to classify the adverse drug reaction based upon this table. So we can also include other classifications according to the researchers of Rowlands and Thompson's, which will include type A medication and type B, according, of course, to their augmented effect, or is it a continuous or delayed one. So all clinical scales should be given according to a certain score, depending, of course, on the side effect that we examined. Is it a life-threatening emergency cases, or is it a simple annoying side effect? Also, we should assess the severity of the side effect. Did it contribute to death or to a life-threatening condition? Did it require certain treatment or no treatment or hospitalization at all? So some medication, for example, can cause constipation. This is considered as a no treatment or hospitalization side effect, where, for example, if it has led to UTI and then it would lead to sepsis, and sepsis would most likely lead to death, then this has evolved from a minor side effect to a more severe lethal effect. Therefore, in certain cases, the impact of the drug will depend, of course, on the dose or even on the human participant themselves. So an allergic hypersensitivity reaction can either be classified also as a simple side effect or as a more lethal complication. So if the drug did actually bind to the target cells or the IgG, then this would have a small molecule that will require a carrier protein, or if it was a larger molecule, then it will bind directly to the IgG. So in this case, it would lead to a relevant immune cell proliferation, and if the drug was used the next time, then the immune system will be primed, and therefore an anaphylaxis would be required as the patient may have certain allergic reaction. So the diagnosis for this case will depend on the skin testing and the genotyping of the specified patient. And the treatment will be through anaphylaxis, which is the emergency treatment, and otherwise supportive antihistamine steroid medications. So how can we actually define the adverse drug reaction in general? So the common adverse reaction will be detected in the clinical trial. This is the ideal timing. And even though that about 10% will be detected after the trial because of the mass implementation. So there are also some rare adverse drug reactions that would only occur in about 1 in 5,000 participants and will not be detected in any trial. So in these cases, they can use the yellow card scheme with case reports or hospital statistics to monitor these adverse drug reactions. So how can we actually identify these adverse drug reaction in a specific patient? So we need to ask the question, is this adverse reaction due to the certain drug, or is it actually due to other factor in this patient? This will, of course, depend on the chronicity of the condition. Is it an immediately relative or reaction, or even is it a delayed, much harder reaction? And we would also need to ask ourselves in the trial, can we stop and re-challenge this medication? Also, can the drug be stopped, or is the reaction allowed to be happening in this stage because it will have a simpler, a minor effect? So in the opinion of the study team, was the adverse event due to the study drug, or was it actually due to other impact or environmental factors? So we need to consider, should we stop the treatment or should we reduce the dose, or even should we switch the class of the medication that the patient is actually consuming? So that's why we need to have careful monitoring during all phases of any clinical trial to ensure that any adverse drug reaction is not happening. So in the first dose administration, we need to minimize the risk with the dose selection and also assessing for the study design. And in the first study in humans, we need to consider what do we need before the study starts, and also the objectives, the responsibility, the risk assessment, and minimization, with also the study population. Are they healthy volunteers or patients? Also, we need to assess the dose and the study design and what we have learned from any previous human studies. So our objectives in the first time in human study should be the tolerability and the safety of the medication, the pharmacokinetics in the relevant dose, and also the dynamics of the effects in addition to the dose concentration, the response for the desired and undesired effect in general. So what do we actually need to do before this type of studies? We need to have satisfactory evidence of the desired activity with the primary pharmacology, with the potency and the selectivity of the drug. Also, we should assess the toxicology of the medication and having any satisfactory safety effect, in addition to the metabolism of the medication and the metabolic enzyme inhibition and induction. Also, we should assess the sensitivity in the specific valid assays and the pharmaceutical formulation from the batch of the known impurity, and also the stability of these manufacturing techniques. So who is in charge or responsible for conducting all of these steps? So many of the researchers who are conducting this clinical trial are involved, and these should include the sponsor, the organization that is conducting the study, the principal investigator, and also the nursing and other staff with the ethical and regulatory authorities. So how can we actually minimize the risk for the humans with the investigator brochure? We should do the preclinical data that suggests there are useful therapeutic window with minor or even no side effect at all. We should have the data that enable them to calculate the starting dose with the responsible certainty, in addition to what are the likely first signs of toxicity and are they even easy or difficult to be assessed. We should also consider, is the dose likely to be limited by tolerability or would it have serious toxicity? Also, if we understood all of these informed consensus and if we should get any expert opinion for certain ununderstood points. So with certain medications, such as biologics and gene therapy, there will be higher risk for implementations. So there are the factors which contribute to this risk, including the quality of the material that is used, and it can be different from that used in the non-clinical testing. Also, these can have side effects on the safety and the pharmacokinetic of a questionable relevance due to the absence of the target effect and also the dose calculation based on the biological activity. So these effects can be poorly understood, in particular if any implications can have or can even happen with multiple signaling pathways. Also, the expression of the target on other genes or cell types or tissue can result in an unexpected biological activity in certain populations. Also, the immunogenicity can result in the hypersensitivity reactions due to the activation and the completions of certain side effects. with the pharmacokinetic that is frequently considered as a non-linear with the target-mediated dispositions. So, to minimize this risk, we should assess the study design and use the preclinical information to select the following criteria. These include the study population, the number of subjects that should be included, representing the sample size, the starting dose, which is of most important, and the dose increment and the maximum dose that shouldn't be overthrown, and also the safety parameters with the duration and the crossover design, in addition to the interval between each dose and the clearance time of the medication with the excretion from the body. So, choosing a principal investigator and staff that are appropriately qualified, experienced, and equipped will help in avoiding most of these issues. And using the appropriate sample size or volunteers will also lead to reducing such side effects or risk factor. We should also recommend an antidote and trained staff in its use due to accounting for any possible side effects which can actually take place. And we should review all of the data before escalating the dose, and it should be prepared to modify the design in light of these data, such as the dose increment and the duration of the washout period. So in regard to choosing the study population, there are some advantages of using a healthy volunteers. These will include having readily available participants who are providing rapid recruitment. Also, there will be no confounding factors such as accounting for disease or other medication intake. Also, it can usually tolerate the pharmacological insult and can accept the intensive schedule of procedures due to their availability, and they can freely give informed consent without having pressure due to certain chronic condition or trying to find a better treatment option. So how do we define a healthy volunteer? This is an individual who is in a good general health and doesn't have any mental or physical disorders that require regular or even frequent medication, and is also able to give valid informed consent to participate in a study. So these can include questions of including females who are pregnant or non-pregnant, the age of the participant, and also their neutrophils, for example, count, and their medication intake. While if we did actually include patient volunteers for assessing the effects of certain medication on a certain disease, we could actually learn more by obtaining information in the patient if they are practicable or even if they are safe. So in a patient expressing the target, the biologics can actually show the target-mediated drug with the disposition, including increasing the dose. Also, the tolerability can be quite different from that in subjects who don't actually express the target. In addition to this, some medications are considered to be too toxic, such as anesthetic or the stimulant of the immune system. So how do we actually select the study population if they were a healthy volunteer or patient? The first thing to consider is that we shouldn't do harm for any participant. So we should minimize the risk and have the most minimal risk ratio ever. And in the case of healthy volunteer, we shouldn't be exposed to anything more than the minimal risk, and therefore not having a highly increased dose, for example, or a highly intervention that would result in a side effect for them. So in phase 1 studies, this shouldn't include any toxicity study, but rather should assess the safety of any medication that is administered. So another major consideration is choosing the route of administration. So in the oral route, these can only include the small molecules because, as we mentioned earlier, this will lead to the absorption from the GIT and the pH of the stomach. So we should consider the variability of the bioavailability, the rate of the absorption, and therefore the Cmax and the Tmax, also the taste and the blinding of the medication, and consider that if we did actually give this medication through an oral route, there is no going back. So for the intravenous route, this should also be small molecules and biological samples and can only be administered slowly, and we can either slow or stop the infusion in the case of any possible side effect. So any route should be supported by the toxicology to avoid any adverse drug reaction. Therefore, after choosing the route, we should choose the doses of that medication. So what is a reasonable starting dose? This is the maximum dose that could be tested, and we should also consider the reasonable dose increment in general to avoid the development of severe side effects. Also, we should consider how many doses that can reasonably be tested in a particular patient, and what is the stopping endpoint or the upper limit of this type of medication. So the stopping point could be the adverse effect point that we should have the toxicity effect observed upon, and the plasma concentration, or even the pharmacodynamic of that medication. So in the pharmacodynamic endpoint, this can include the maximum enzyme inhibition, the receptor blockage, and even the occupancy. And for the maximum recommended starting dose according to the FDA, step 1 should be including determining the no observable adverse effect level, which is the NOEL that we have discussed previously yesterday. Also, in step 2, we should convert this NOEL to the most appropriate species to the human equivalent dose. And in step 3, we should select this HPD dose and take into account any additional factors, including the metabolism, the receptors, the blinding, and also the most sensitive species. And in step 4, we should apply the safety factor, which is a fixed factor for most of the medication, and therefore assess in step 5 the adjustment in this medication upper limit on the pharmacologically active dose. So what are the bases for the dose selection? This includes the pharmacology, which can actually range of the doses at which the desired effect is seen in the in vitro or in the in vivo study. This will be in animal model studies, and the relevance of these models in a human subject should be converted into the NOEL ratio. Also, the range of concentrations at which the desired effect is seen in the animal models and in the relevant models to these ones should be taken into consideration upon the decideness of the dose. So as we can see here, this is the dose response curve, which will have multiple points according to the slope and the estimated drug action with the efficacy point. So from a pharmacological point of view, we should assess the nature of the pharmacodynamic activity in the animals and the slope of the dose response curve, as the ranges of the doses at which the desired effect is seen should be assessed with the rate of the concentration in the in vivo animal models with the relevance to models in human disease. So there are some basic medications affecting certain systems in the body which should be assessed for extra safety measures. These include medication which affect the CNS system or the respiratory system, or even in the case of renal impairment, as we mentioned previously. So it is to be expected that doses producing undesirable effect in the animal will be lower than those of those producing the target organ toxicity and therefore should be monitored carefully in human subjects. So the toxicology should also be assessed by assessing the plasma concentration and the exposure at the maximum no adverse effect level. This is the most sensitive toxicology species and need to be decided with the parameters that is most important. Also, the nature and the severity of the toxicity that is seen in animal models and in the exposure at which the toxicity occur should be assessed. So it is necessary to assess the consequences without assuming that the bioavailability will be 100% in humans. So in the single ascending dose study design, this should be randomized and placebo-controlled double-blinded study. But how can we do this and why? This will help us in allowing the intra and the inter-individual crossover with comparison of the adverse effect and also the pharmacodynamics. In addition, it will allow or even it will avoid the bias in conducting of the study and the attribution, in addition to the handling and interpretation of the adverse drug reactions. Also, the statistician in this case can generate codes for these data to be entered and analyzed later on, and it can be available to the individuals who require it during the study, such as the pharmacokinetic information for assessing the dose. So each individual or subject who are participating can affect the study design and can only have a new cohort that is treated at each dose level, therefore shouldn't be participating in more than one study to avoid any misinteraction of the study medications. Also, each subject participating on several occasions with the same cohort treated at various doses level using a crossover randomization will help in assessing the design. And anyone who participates on several occasions with the same cohort and even with different level of the parallel group randomization need to be assessed for any side effects. So if we have a new cohort for each dose, then we would need to divide them according to the following table, with having an example of randomization of the study cohort participant. And it will have an advantage of no problem with the dropout and even doesn't need breaking the blind or carrying out overdue to the long time with the limited blood samples, for example. But of course, it will have some disadvantages of uneconomic with many volunteers that are screened and also no possibility of intra-subject comparison due to the large sample size. Where in the case of the multiple single-dose crossover studies, this will have multiple subjects participating in 4 different studies, and in this case, the randomization is restricted and also can help in preventing any subject from actually receiving placebo on a successive occasion. So we will be taking a break now for Salat al-Isha, and then we will continue with discussing the different type of designing studies with medications. Assalamu alaikum wa rahmatullah. Welcome back, everyone. So, as we were discussing, the appropriate doses of clinical trials will be also determined by many factors. One of these factors include the dose. So, to base the decision for the dose escalation or the selection of the dose for the next study is of paramount importance. And we should be sure about the linearity or the pharmacokinetic, and also to give reliable eliminations of the PK and also any other parameters. In addition to this, to give a reasonable estimated of the pharmacodynamic is also of paramount importance. And also to meet the study objectives without putting unnecessary numbers of a subject at risk is also of paramount importance. So the dosing intervals is also of very specific importance in most of the cases, as we need to ask ourselves how many subjects should we give a dose in the first day, and also at a new dose level, we should reduce the risk by the staggering exposure. So if we, for example, had a cohort of 9 subjects, we can randomize it, for example, in a dose of 2 to 1. This will include, for example, 1 active, 1 placebo on day 1, and then in day 2, we can have 5 active and 2 placebo. So what the intervals should be, or should we leave between these dosing of the different subjects at particular dose level? This will, of course, depend upon the time of the profile of the activity of the dose. This is the Cmax that we have explained previously. So also we need to consider what interval should we leave before administering the next dose or even during the washout period. This will, of course, depend upon the time that is necessary or needed to analyze and review the data, and also the half-life of the drug, and also of its metabolite that need to be considered at once. So an example of this is combining the single and multiple dose study in a duration that is over 1 year. This will, of course, depend upon the participant we have, but this is a simple example that we can use. So what else can we include in this type of studies? This can include the fit and the fasting status of the patient, and we can compare them accordingly. Also, this includes the age of the participant, if they were elderly or even young participants. In addition to this, this can include the solid dose from form or even the liquid formulation of certain doses. Also, this can include if this was single or repeated dose, or even in the case of repeated doses, we should assess the tolerability and the safety of every medication that we are administering. So the data from the exploratory studies in male participants found that regarding the tolerability and the safety, this can include the adverse event and also the adverse reactions in general, also the vital signs calculations, and the hematology, the biochemistry, and the urinary analysis, and other forms of effects that the medication can actually have. Also, the pharmacokinetics can be assessed, including the relevant dosage range in regarding to the Cmax, the Tmax, and the half-life of the drug. also the dose proportions and the concentration of the major metabolites in the plasma, in addition to the amount of the drug or the metabolite that is actually excreted. Also, the pharmacokinetic can be assessed in addition to the dose concentration response, which is of paramount importance in this case. So, in conclusion, the safety of the volunteers is of paramount, and we must always minimize the risk by anything more than minimal risk that is unacceptable for any healthy volunteers. And we can have three varieties of study design that are suitable for human involvement in this type of studies. And these risks can be minimized by taking into consideration the account of the nature of the molecule and what we want to understand about the mechanism of action. Also using the most appropriate study design in addition to the rational selection of the starting dose based on the pharmacology and as well as the toxicology. We need to also ensure those conducting the study are trustworthy and able to report any side effect. In addition, the monitoring of all safety factors during the study and modifying the procedures if necessary is required. So there are many implications for pharmacy and clinical research, including many, such as the manufacture and the supply of the drug, the clinical trial regulation, and also the other regulations such as the clinical trial drugs and septic of the medication, and also many other pharmacy site files that can be included with this type of medication. So the investigational medicine products can be included as the pharmaceutical form of the active substance or even of the placebo that is being tested. This can also include the placebo used or the reference medication under consideration. Also, an unlicensed medicine or medication which indicate that the investigation aspects of a drug with license need to be conducted in addition to the assembly or the inclusion in the container, which is labeled before the product is even sold or supplied to many pharmacies or supplies. In addition to the reconstitution, which is dissolving or dispensing or even diluting the medication with another substance or even using a vehicle for the purpose of administration. So, good manufacturing practice can cover the equipment, the materials, the documentation, the quality control, and the quality audits, all of which can be included in this INB. So, it needs to assure that the product is of consistent good quality. It needs to assure that the development of the data on which the license is based is actually valid. It needs to hold an actual license for the medication with quality products and even labeling to the regulatory bodies and sponsoring the responsibility, but also having assured compliance of the INB. In addition to this, an active pharmaceutical ingredients should be assessed, including the chemical entities that are already existent in many manufactured medication, and having the Europe government regulation objective is considered a good manufacturer to ensure the quality and the purity of the medication. So the final product labeling is also of paramount importance due to the labeling legalization and that everything of the trial information need to be legally recorded. So there are some quality products that need to be filled out and mailed to the procedures or regulatory bodies, including these identification of the product component and also details, which must be included with every study. So in the clinical trial regulation, these can be different in different authorities, bodies such as in Euro or the UK. And of course, these will be different in KSA depending on the type of research that we are conducting. So the legalization development in the Euro was developed wide for developing or enabling the regulations to be having the targeted implementation to keep the patient safety. So the key implications for pharmacy included regulatory submission, having low interval trials, and even having a recommended age range in the participants with even having archives. So these will be answered depending on the type of the trial we have, the clinical trial, with also the type of the subject that we are involving. So this figure can be very helpful in answering this type of questions. There are, of course, other regulations for non-clinical trial studies. These are the studies which are not classified as clinical trial medicine under the assumptions of the MHR, which will involve giving any medicine that must be still managed according to all other legalizations. These can involve the phases in which the drug is being used for their known effect to trigger a response, such as in a particular receptor, so it can be scanned in a different population. Also, these can include drugs that are used as devices for studies, and also the blinding oral drug in the in-house by overcapsulations in the special license populations. Also, these can include or involve the CTIM and also the non-product license that should be produced. So the legalization for the non-IMB can include Medicinal Act regulations, and then this was developed to the Medicine for Human Use regulation and the Human Medicine regulation in 2012. So in any clinical trial involving drug, there can be some problems. So the pharmacy is responsible for all drug in the hospital, and in the trial drugs, these can have some more problems due to the fact that these are new formulations, and also these are less suitable with having less data and may not be available for or even patient ready in general. It can also need to prove the efficacy and the safety with ensuring the safety, the correct strength, and even provide or maintain the blinding. In addition to this, good clinical practice regulation complex can open the interpretation in this field, while also having other regulations that applied to pharmacy. So there are three key areas to manage IMBs, which are ensuring the source, the accountability, the storage, and also the preparation with dispensing or even aseptic compounding. So we should have unique drug design that doesn't use the charge for supply. And occasionally, in these cases, they may be used for administration if there are multiple inpatient doses which are needed. So in the dispensary, all IMPs are ready to be taken by the patient for a simple reconstitution of oral therapy or even of IV. In addition, this can include labeling and also of the patient details. So there are an electronic prescribing system that can be used for some medication in the inpatient out or even in the outpatient setting and can also be applied in research or some other type of pre-dispensing machines such as the aseptic type dispensers. So the aseptic service in clinical trials include a trial-specific prescription that is received in the pharmacy, also the clinical screen by the pharmacist against the trial protocol and the patient's blood result. In addition to any prescription changes that are needed or even made by the prescriber, and the prescription that is rescreened, or even the ones that would need entry into the pharmacy computer system, and also the final product worksheet and the dispensing labels that are produced. Also, these can need assembly of the drug and the consolidation of the drug and details of each medication that are entered into the worksheet. These can also include mandatory accountability logs and also process check of the assembled worksheet and the completed accountability logs by an accredited checker. So if we have a clinical trial and we are using new drug or a placebo, then we can have this form to guide us in the required dosing or the manufacturing information that should be included in the study. So the clinical trial prescription process in the aseptic service can include the aseptic compounding by an accredited and validated member of staff, also all volumes that are double checked, in addition to the infusion or injection product, which is the final checked and labeled in accordance with the legal requirement by accredited final checker. Also using IMB and vials that are discarded as per the Trust Health and Safety Policy, with the final product that is released from the pharmacy to a member of the nursing staff for the administration to the patient that are included in the trial. So all of these steps can actually take very long time to be implemented, but these are of paramount importance for the assurance of the quality and the safety of any clinical research trial. So the procedures can include the SOPs for the clinical trial management in the pharmacy, such as trial temperature monitoring or any emergency code break, in addition to a trial-specific procedure in every aspect of the handling of the trial. In addition to the staff training, including induction and accreditation for non-clinical trial staff, so they can actually dispense the trial and aseptically compound them or even unpack and receive the study drug. Also, the medical and the nursing staff training in the IMP management and out-of-hour helps can be important. With other types of trial, including gene therapy that would need training, occupational health clearance, and designated with a biosafety cabinets. And in the non-clinical trial studies, this shouldn't be covered with the CT regulations and can still be considered as blinded type of studies and need to be implemented accordingly. And in regard to the internally sponsored studies, these can include working with the investigators and the R&D research offices to facilitate any IND procedures to ensure that regulatory compliance can be included. So these can include sourcing the drug and arranging the manufacture importance, and also sourcing or matching the placebo for blinding trials that can be real difficult to achieve in most of the cases. In addition to, if there was septically compounding of the placebo or active in-house, then the pharmacy will be unblinded when the study teams are actually not, and therefore these guidelines should be followed specifically to avoid any complication from being developed. In addition to this, the technical agreement with the manufacturer to cover the dose and when not to cover it is important to be included with the quality agreement and the qualified personnel's release in the drug, with any application being completed and labeling the medication and having a green light for the product release for each shipment before it is actually dispensed. There are also other activities that need to be assessed, including the ethical committees, as each one has to have pharmacists as one of the experts alongside lay members. And in the case of gene therapy, there will be a GM committee with multiple disciplinary committees and governing all aspects of gene therapy to ensure that the legalization and guidance are being followed. And in addition to this, there should be clinical research facilities that will facilitate the protocol review and the quality with the safety committee being followed. In regard to the funding, we should ensure the cost and neutrality of the commercial trial is actually being reasonable and ensuring unsupplied study drugs in other studies that are funded from somewhere and are offset by the saving or the standard treatment cost. In addition to ensuring that the safety of the financial reimbursement are actually being carried out for the patients. So in most hospitals, the pharmacies will have dedicated clinical trial staff, plus other staff who are trained to dispense, compound, and even receive study drugs. So which one do we need? This will actually depend upon the process or the study drug that we have. So in a sponsored trial coordinating center, the pharmacist will check the clinical aspects of the trial involving in the writing of the drug protocol and checking the excess treatment costs. They will also be involved in the dispensing of the process with otherwise pharmacy technicians that can perform functions including setting up the trial of the site and also the sources of the information and manufacturing with managing the site file and training the non-specialist staff. So in the pharmacy site file, This can include the essential documents, depending, of course, on the stage of the trial and what will be in that file, including the pending, the active, or the closed information. And a lot of these documents will even be duplicated with the TMF, but can or must be held for a file that is to explain the process, even for future events that we need to look for. So the trust sponsor would have additional requirement to those stated below, and the pending document, when they are first actually proposed at the site, can include the protocol, the contact detail, the special requirements, the storage and handling, the drug supplies, and also the excess treatment costs and consideration, which need to be taken into consideration for the study drug. In addition to this, these can include the investigator brochure, the quality control release, and the assurance release with labels of the medication and any prescription or worksheet, in addition to the accountability of the drug reconciliation and returns, with any consumable to be used to prepare the injectable doses, with agreeing to the destruction procedure per the site protocol, and also unbinding arrangement of the IVRPs and the clinical site agreement, and also the ethical approval, the sponsorship level, and any other important information that need to be included in the file of the study. Also, regarding the active site file that include the active information, such as the trial-specific procedures, the protocol amendment, and any new versions with the summary of change, even if it doesn't affect the pharmacy directly. Also, all approval for current versions of the protocol, and if any amendment affects the drug that can or need to be changing in the prescription, and also the worksheet or storage. Also, the signature log for all pharmacy staff who work on the trial, with patient log and listing all enrolled patients, or even completing the prescribed information, in addition to randomization with drug delivery and also temperature excretion report. In regard to the closed information, these can include confirmation of closed down, including the agreement to break the blind if it was required, or even confirmation of the drug dispensing, including destroying or returning the drug in general. In addition to confirmation of the code break disposition and even the final invoices of the medications, with confirmation of archiving arrangement, and also having archives with the investigator's file that should be included at all parts of the study. So another important consideration is the good clinical practice, GCP. This is important in every step of the research process to ensure that every guidance or step in our clinical trial is actually with accordance to the patient safety in mind, without having any negative impact on them for participating in the study. So the good clinical practice is an international ethical and scientific quality standard for designing, conducting, recording, and even reporting the trials which involve the participation of a human subject. So these can be helpful in providing the assurance that the data and the reported results are actually credible and accurate, also assuring that the rights and the confidentiality of the trial subject are being protected. So the ISGCB was amended in 2016 as the last amendment for this protocol, and it included or consisted of the principle with the Declaration of Helsinki that we have explained previously, and also the unified standard that provide the mutual acceptance of the clinical trial data by any regulatory authorities, with the harmonization in four areas, including the safety, the quality, the efficacy, and even in the multidisciplinary approaches. And in regard to the efficacy, Section 5 was amended to even from 6 to 13 principles of the good clinical practice in general. So the International Conference on the Harmonization of an Organization of Regulatory and Pharma Industry to discuss any scientific and technical aspect of the process can be found in this link. So the Tsuji syphilis study was one of the first studies that included or set the basis for the good clinical practice in human trials. So this included 40 years study of 400 African American men who had syphilis in the USA. This was conducted by the US Public Health Service, and the subjects were told that they had bad blood and were followed up until death without having any administration of treatment, even though that there was an active treatment that was recently discovered, penicillin, but the study participants were denied from having access to this treatment due to their participation in the study, and therefore resulted in their death. So as a result of this had the Belmont Report, which issued 3 ethical principles for research, which is the respect for the persons, the beneficence, and the justice at all steps of the research process. After that came the case of the Nuremberg Code in the 1950s, where the voluntary consent of the subject included or resulted of a fruitful and not proceedable by other method. And they stated that the trial design should be based on the result of an animal experimentation first, and they should avoid unnecessary suffering upon the participant. And also, if the researcher believes that the death or disabling injury can occur during the trial, then the research is not legitimate and shouldn't be done at all. Also, the degree of risk may not exceed the benefit of the experiment at any point. and every possible action must be taken to protect the subject from injury, disability, or even death. Also, the experiment should be conducted by a scientifically qualified personnel, and the subject can terminate the trial at any stage, with the investigators must be prepared to terminate the trial at any stage that any side effect or harm did occur to the patient. After that came the directive law and the guidance with the Euro regulation of 2014. This replaced all the clinical trial regulation across Europe and increased the harmonization with a single authorization for the recommended research guidelines in the UK. So the safety reporting has changed across the world with simplifying the requirement for the low risk trials and clearly archiving about 25 years of research and the discrepancies in any other projects. So there are some principles of the good clinical practice, which included that the clinical trial should be conducted in accordance with the principles of the Declaration of Helsinki, the GCP, and any other regulations that is being followed. In addition to this, the anticipated benefit for the individual and the society should outweigh the foreseeable risk and the inconvenience for any study participant. Also, the rights, the safety, and the well-being of the subject should prevail over the interests of the science and the society. So the patient safety is of most important at all times. In addition, the available non-clinical and clinical information should be adequate to support the proposed trial, and the trial should be scientifically sound and described in a clear, detailed protocol. Also, the trial should be conducted in compliance with the protocol, which has ethics committee approval that is of a very favorable opinion. Also, the medical care and the decision should always be the responsibility of the qualified physician who is in charge of the study. In addition to this, each individual involved in conducting the trial should be qualified by education, training, and also experience to perform their respective task. And they should be freely given informed consent that should be obtained from every subject before the clinical trial participation. In addition to that, allowing all the clinical trial information that need to be recorded, handled, and stored so as to allow its accurate reporting, interpretation, and also verification irrespective of the type of the media that is being used. Also, the confidentiality of the record must be protected in accordance with the regulation, and the IMPs should be manufactured, handled, and stored in accordance with the good manufacturing practice and used in accordance with the approved protocol. So there is another regulations of the good clinical practice that include additional principles from the UK trial regulations. These included the right of each subject to have a physical and mental integrity that must be safeguarded at all phases of the study, regardless of the impact that may have on the study itself. in addition to the licensing authorities' approval that is required before any trial can be started. And the protocol must define the inclusion and exclusion criteria, the monitoring and the publication policy, in addition to the provision that must be paid for insurance and indemnity to cover the investigator and the sponsor liability. So, what is a clinical trial of an IND? This is any investigation in human subject other than the non-interventional trial that is intended to discover or verify the clinical, the pharmacological, or any pharmacodynamic effects of one or more medical products. This also needs to identify any adverse reaction in one or more such product, and also to study the absorption, the distribution, and also the metabolism with the excretion of one or more of such products. And with the objective of discriminating the study safety or the efficacy of those products, this is the main consideration in these types of study. So there are many research governance that can be included, including the systems or the regulations which ensure the good practice and high quality at all stages of the research process, including ensuring that the researcher do the right thing in the right way at the right times, and not having new legalization but the existing one into guidance to guide the clinical research process. So the NHS can have many research governance for the purpose of ensuring the dignity, the right, and also the well-being of the public, and to improve the research quality or even to prevent the poor performance and adverse events, and to help prevent the misconduct and fraud in the research, with also covering the entire research process, including the design, the management, the undertaking, the funding, or the participation in the research itself. So the policy framework for the health and the social care in 2017 has focused on the patient, the service user, and the public. This included a safer, more efficient, and more effective treatment and care, with other services to be developed and tested for the sake of the patients. This helped in applying to the research to be less barbaric and more focused on the patient safety. And the industry sees, for example, that countries following this regulation as a place to do health and social care research. So these research projects that are registered with data and tissue to be made available for future analysis, research finding, can get even published and summarized for those who took part in them. So the policy of this framework included 19 principles, such as the responsibility, which are detailed for everyone participating in the study, such as the chief investigator, the research team, the student and the supervisors, the funders, the sponsors, and basically anyone involved in the study. And as such, the good clinical practice covers the documentation process, including the protocol, the trial record, the approvals, and any amendment with the recruitment, the consent, the safety reporting, the monitoring and inspections, with even the archiving of the study data. So the safety reporting is considered as a legal requirement in any study, as stated by the Medicine for Human Use, as any investigator shall report any serious adverse event immediately to the sponsor. So the sponsor shall keep a detailed record of any adverse events that are related to the clinical trial and are reported to them by the investigator of that trial. In addition, these can include the annual report, as there are two types of reports that need to be submitted during the trials. These include the annual progress report and the development safety update report. And at the end of the trial, they need to give an end of study notification with final report within 12 months. So the annual progress report is submitted to the REC on the anniversary of the original ethical approval, and the update should include the site information, the recruitment, the amendment, and also the safety. Where the development and safety update report should be included or replaced by the annual safety report in the MHR in 2011, and these were introduced to improve the harmonization across any participating research country, and have comprehensive annual safety review with the investigational drug, with even having sponsor responsibility that is dedicated to the chief investigator. So after the trial completion, there is another set of documents that need to be sent, which is the end of study notifications. And this should be sent out within 90 days of the study end date, and also within 15 days if the study was early terminated due to any source of issues. And the final study report is sent to the responsible authorities within 1 year of the end of study notification. After that comes the archiving of the study. These should include or keeping a log of all documents and where they will be located, in addition to ensuring that the data and any software is retrievable at all points. Also, they should be labeled on the medical notes to be retained with electronic record and software record that need to be carefully archived. So how long do we actually archive? The local requirement, for example, 10 years after the end of the trial, is different depending, of course, on the source of information we obtained from the study. This can be at least 3 years after the last marketing authorization. And if the trial involved children under the youngest participant, then they should reach the age of 21 years before these archives can be discarded. Also, at least 30 years for any advanced therapy trials, such as gene therapy, should be recommended. But there are some new regulations in place that it will be 25 years only, depending on the type of the study. In addition to this, the safety reporting is considered as a legal requirement for most studies, as an investigator shall report any serious adverse event immediately to the sponsor, and the sponsor should keep detailed record of all adverse events that are related to the clinical trial, which are even reported to them by the investigator for that trial. Thank you for your time. We have reached the end of our webinar for today. If you have any questions, please do send them on the chat so we can discuss them. العفو، بالتوفيق جميعا. الله يعافيكم، بالتوفيق للجميع.